Hey guys, so I wanted to go over the notes from Friday, January 22nd, the microscope introduction PowerPoint. So I wanted to go through that and go through Schoology with y'all because I know it's going to be a little different with me not being there. Okay. So on Friday, January 22nd, you should have gone to the Schoology page and watched the Amoeba Sisters. I'm going to share my screen with you now. Sure. Okay. So I'm going to go, this is the Schoology page. Okay. So you'll scroll all the way down to Unit 3. That's where we are. So January 22nd folder. So this is the strange teaching video. This is the video that you are watching right now. Okay. So this is me telling you, basically, I'm going to go over the notes today with you and the interactive notebook pages. Okay. Just like I would if I was in class. So you would go to microscope information first. Okay, this is the Amoeba Sister video I need you to watch. Okay, it's 9 minutes. It's not terrible. Okay, basically she's going to tell you how these scientists looked at cells, which cells are microscopic organisms, right? And so they needed microscopes to look at small organisms. So anything under a microscope is going to have to be super small, right? Because micro equals small. And so basically they're going to tell you how these scientists came up with the idea that, hey, there might be something smaller in water, or there might be something smaller in matter. So basically how they looked at it under a microscope. Okay, so watch the Amoeba Sister video and then come back to this video. Okay, I'm going to give you a second to pause it. So 3, 2, 1. Okay, so now you've watched the Amoeba Sister video. So you're going to go to the microscope PowerPoint. Now I'm going to download it and I'm going to show you how you can download it, and you can rather pull it up and flip through it with me and kind of have like me on half your screen and then the notes on the other half of your screen. Or you can just leave me big, and because you can kind of see like my face small and you can really see the PowerPoint, and you can just pause the video, write down the notes, and then listen to me talk and write down like the little analogies and the things that will help you remember what I'm saying also in your notes. Okay, so up to you. I'll give you a second to do that. All right, I'm going to get my screen set up. Okay, so now I'm going to go through the PowerPoint notes with you just like how I would if I was in class. Okay, so microscopes. First slide, we're going to talk about Robert Hooke. So usually I tell you that there's like a bunch of old dudes and you don't have to know their name. These people, you have to know their name. Okay, so Robert Hooke was the first guy to identify cells. Okay, so he built the microscope. All right, so that will be a question on the state test. It was on the state test pre-test questions. It's in the textbook pre-test questions. It's on our unit 3 test. Okay, who built the microscope? Who was the first to identify cells? Robert Hooke. Okay, it's kind of like in English when you're supposed to start your essay with like a hook and key. All right, so Robert Hooke built the microscopes. He was the first to identify cells. Okay, how did he do that? He looked inside a cork, like a cork, like at the very top of a whiteboard that you can like stick a thumbtack into. Okay, kind of like a cork board. So basically he looked at that and he could see pores, kind of like if you like look at really deep, really like close into your nose, and you can see like your deep skin pores. Okay, basically he saw that and he was like, I bet you I could see something smaller inside. So he built a microscope to allow him to basically zoom in on that piece of cork. Okay, so next up is Antoine Lon Van Wyck. Okay, totally butchered his name. You don't know how that, you don't know, blah, 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 blah. You don't have to know how to say his name. All you have to know is that he was the father of microbiology. So Antoine was the father of microbiology. What is micro? Micro is small. What is biology? The study of life. So microbiology, this is the study of really small living things. Okay, so basically everything that you have to observe under a microscope is going to be microbiology. Okay, so next up is exploring the cell. So basically, these are the different types of microscopes. Okay. So it says most microscopes use lens to magnify an image of an object by using light or electrons. So on the microscope right here, the light source is rather going to shoot light or shoot electrons through your specimen in order for you to view it. Okay. So two types of microscopes. So in your notes, I want you to write right here, number one. So I'll kind of put it in so number one, light microscope. Number two, dissecting microscope. Okay, so the two types of microscopes. So types of microscopes, right? Compound light microscope, okay, which all you really need to write for that is that under optimal conditions, it can magnify objects from 40 to 2,000 times, okay? So I want you to know this 40 to 2,000 times. You don't have to memorize that, but you need it in your notes for the test, okay? So basically this specimen's original diameter using the light, so it can go from 40 to 2,000 times. So it just zooms in that, that amount, okay? This uses light that goes through it. So a compound light microscope sends light through the object. So you can just say light through the object. Basically, when you shoot light through the object, it doesn't destroy it. If you shoot electrons through it, it's going to end up destroying the specimen because the electrons are too powerful. Okay, so that goes with compound light. So now your next one is dissecting light microscope. So you're going to write that and you're going to say it looks at larger items. Okay, this one is looking, compound light is looking at smaller. Okay. And then dissecting light is looking at larger. Okay. And then I want you to know that it goes from 20 to 40x. Okay. So after you got that down, to the next slide. Okay, so this is a picture of the compound light microscope. Okay, so basically when we are in the lab, when I'm back from quarantine and we're not doing this virtually, then this is what your microscope will look like. Okay, so we have the ocular lenses. Okay, we're going to get into it in our notes in a minute, but the ocular lenses are going to be 10x. So this is basically a lens on its own, and then you have these additional lenses that allow you to zoom in smaller on the microscope image. Okay, so these are your eyepieces, so your ocular lenses. Okay, this is your nose piece, and it allows you to turn the microscope like your eyes. So like if you and your partner are right here, you can keep the microscope steady, but you can turn it and like your partner can look and then turn again and you can look. So it's just kind of cool for like sharing. Okay, so then you have your objective lenses. All right, so that's going to be your 40x, 10, your 4x, 10x, 40x. Okay, your slide holder. Okay, your slide holder is where it's going to be like a little clip that you can put your glass microscope slide onto. All right, and so it basically just holds it in place on top of the stage. So the stage is where you actually, it's like what you set your slide onto. Okay, so your carrying handle, this is like the arm. Okay, so this is like the arm of the microscope that you're going to carry the microscope with, huh? Okay, so then underneath your stage, you're going to have your condenser, which is also called an aperture lever. Okay, basically this is just going to condense the amount of light that you're allowing onto your specimen. So it's kind of like if you have a like right now I'm in a square, and so it would just be like right now you can only see my hand, but now as I choose to open up the condenser, I would allow more light, more of the image to be seen. So basically that's what a condenser does. Okay, so then you have the light source. Okay, so that's rather going to be light or electrons. All right, and then you have your coarse adjustment and your fine adjustment. So basically, just like as you would use a camera to zoom in and zoom out, your coarse adjustment is one's going to move the stage up and down, and then one is going to basically make the image fuzzy or not fuzzy. Okay, so it's just going to help with the texture. So then you have your light intensity control. So that's basically turning up your brightness on your phone or not. So turning up your brightness on the microscope. So for that, you really didn't have to like take notes on, but that was just kind of like a brief explanation of each part of this microscope, of each piece of the microscope. Okay. So care of the microscope, you're going to clean the microscope using lens paper and alcohol before and after use. So the eyepiece and the objective, so the where you put like look into the eye lenses, okay, the to the eye lenses, okay, you're going to look into them. You have to clean them with this special paper. So this paper is called lens paper because if you blow your nose with tissue like over and over again, you're going to get a little red nose. So this tissue paper, this lens paper, doesn't have like this grit to it, okay? So basically you have to use this special paper. We're always going to store the microscope with a cover just so dust and other particles don't get into it. So all you have to write down for this is care of microscope, clean microscope using lens paper and alcohol, store, I would just say store with a dust cover. Cool. Next up. So you have your objectives. So you have 3 objectives, your 4x, 10x, and 40x. So your 4x is your scanning power. So this is going to be the first one where you always start. 10x is low power. This is going to zoom you in basically just a little bit more. And then 40x is your high power. If you go anything more than 40x, you're going to need to use immersion oil. We will not use this in biology this year, so I'm not worrying about it. I just want you to know these 3 scanning lenses, okay? I mean, objective lenses. So as you increase your power, the field in view gets smaller, okay? So as you like basically zoom in, you're zooming in on the picture, right? So you're not going to be able to see the whole house. If you zoom in more, you're going to see more of the window. And then if you zoom in more, you're going to see inside the window. So it's kind of like as you zoom in, your view gets smaller because you're zooming in deeper, okay? So your total magnification is going to be the objective lens. So the lens that you're putting your eye up to and looking through, and then your objective lens. So your eyepiece and your objective lens is going to, so you multiply those together. So if your eyepiece lens is 10x and then your objective lens is 40x, okay? So 10 times 40 is 400. So then your total magnification is going to be 400x. Okay, so it says keep both eyes open while looking through the microscope. So basically, this is it's kind of like how you want to with binoculars. Like you don't want to close one eye because then your frame's going to be a little bit off. You want to keep both eyes open. I promise it won't be weird. It will, but everything will look normal. Okay, you'll see that when you get into the lab. Total magnification. So this is kind of what I was talking about, okay? So if you do 10, so what would be the total magnification of this? You would do 10 times 100 equals 1,000. So your total magnification would be 1,000, okay? So making a wet mount, so just like titled this wet mount, all right? And so to make a wet mount, I basically just want you to redraw this picture right here, all right? So to make a wet mount, you're gonna have your glass slide. Then you're gonna place a drop of water on your glass slide. And then you're gonna have your little glass cover slip. It's like this really itty bitty little piece of glass. And you're going to put it down at an angle. So if here's my pen and my hand is the glass slide, you're going to put it down at an angle and then let it drop onto it. So you basically minimize your air bubbles. Okay. So just redraw that image and put in, and then you can type this note: add cover slip at angle equals less air bubbles. Okay. So redraw that image and add that bullet point. Okay. So everything that you see in a microscope is going to be upside down and backwards, okay? So basically that just means if you're moving something to the left, you're actually kind of moving it up and down and to the right. So it's going to be funky and you're going to have to play with it. So you don't have to write anything from this slide. Just know that everything, it's kind of like your hand-eye coordination is going to be weird like a video game, okay? So you're going to have to play with it, and it's you can't just base it off intuition. So staining slides. So stains are used to add color to specimens that you want to look at in the microscope to see them better. So write staining slides, stain. So then just write add color to specimens. Okay, so if you're going to add a stain to a wet mount, here are the steps. So one, you would place a paper towel on on the table. Then you would basically put your slide there. Then you would pipette a drop of stain on top of your specimen and let it kind of be absorbed, and then put your cover slip on, and then you would view your wet mount. So you would kind of you don't have to write down these steps. I just want you to know that your staining slides mean to add color to specimen um, so that you can see them better. Okay, so just have that first bullet point down. Okay, electron microscopes. So there's two different types of electron microscopes. Okay, you're going to have a transmission electron microscope and a scanning electron microscope. Okay, so electron microscope, two types. You're going to write this down. One is transmission. Two is scanning. So transmission is a form of electron microscope in which the image is derived from electrons which have passed through the specimen, and a particular one in which the whole image is formed and rather once than by scanning. So transmission, basically it takes a picture. As it shoots the electrons through, it immediately like snaps a picture of it, and then the specimen's dead, so you can't use it anymore, but now you have this really awesome picture. Okay, so so I want you to write transmission. We just wrote that it's a type of electron microscope, so we know it's in a form of electron microscope, so which an image is derived from electrons. Okay, that's what I want you to write here, because then next we're going to write scanning. Okay, because scanning is also an electron microscope. We know this, we've just written this. Okay, in which the surface of the specimen is scanned by a beam of electrons that are reflected to form an image. So the specimen is scanned here. Okay, where here they're just passed through the specimen. Okay, so the bolded points, that's what I want you to write down. All right, so basically I want you to, after you write these 2 points down, I want you to star that the big takeaway from this is that electron microscopes, they're going to destroy the items that are placed in them. Like our light scanning microscopes that we use in the lab, we can reuse those samples over and over again because we're just shooting light through our specimen. But electrons, when you shoot electrons through, it damages the specimen and you can't use them again. So electron microscopes destroy the items placed in them. Okay, so basically it's going to destroy your specimen. Okay, so that's why we can magnify them a million times, because we can like zoom in on the dead specimen and the image over and over and over again, but we can't reuse that living sample. Okay, so this is a basically an image of an electron microscope. You would see this at like Le Bonheur, hospitals, colleges. They have, but they're like super expensive. They're like $500,000, like expensive, like, and they can like go way up. So this is like big bucks, not what we would have in our lab. Okay, micrographs. Micrographs are a photo of an object seen through a microscope. So literally, you can write this bullet point, or you can say micrograph. Let me move for you. Micrograph equals pic of image through microscope. Okay, so basically it's a picture of what you see through the microscope through the computer. So basically you have a camera attached to your microscope and the computer. And instead of you looking through, your camera's just like click, 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 just taking pictures of your specimen. So it's pretty cool. So I just want you to know that micrograph, what's the definition of that? It's a picture of the image that you've seen through the microscope. And these are pictures. This is a micrograph example of yeast. So all of these are yeast, and they're seen, these are different images from different microscopes. So this is a light microscope. So this is like what we would see in the lab. Okay, this is a transmission electron microscope, and this is a scanning electron microscope. So these are all yeast, but it's just cool that you can see different things examining the same specimen under different microscopes. Okay, so cell size. So cell size, certain units of measurements are used for tiny objects such as cells. So basically cells are itty bitty, right? Let me see if I can zoom in. So we have nanometers are basically where cells start out as, and each cell has different units of measurements. All I really want you to know is that DNA is going to be less than 1 nanometer. So I'll write this out for you. So an example of cell size. So certain units of measurements are used for tiny objects, cells. So this is exactly what I want you to write. So example, DNA is less than 1. Okay, so that's what I want you to write for that. And cool. So that is the last slide of notes. Okay, so I am going to now change to my interactive notebook page. Okay, so now I'm going to go over these pages with you. Okay, it's just page 1 and 2 today. So go ahead and get out your unit 3 notebook while I get out mine. All right, and so we're going to do these together. All right, so you have this little word bank over here. All right, I am going to basically delete the word answer on my text box and then type in what the correct answer is. So what is this top arrow pointing to? Okay, this is going to be the arm of a microscope. Okay, what are the top pieces that you're going to put your eyes up to? Okay, these are going to be the eyepiece. Okay, then this is the nose piece that you're going to rotate, and these are going to be your objective lenses, okay? Okay, this is going to be your stage. Okay, this little tiny itty bitty lever is going to be your condenser, and your condenser is, condenser. So your condenser is going to be your, basically how much light you're allowing in to the microscope. Okay, so then we have the diaphragm. Okay, so the diaphragm is also helping the condenser adjust light. Okay, so then down here is going to be your mirror or light source. Just like in the classroom, guys, can't spell and can't type, right? Wow. Okay, so now we're down here on the bottom. Okay, so this is going to be the base, base. Okay, so now these are going to be your coarse your adjustment knobs. So one of them is going to be your coarse and one of them is going to be the fine. Okay, so think about it like the alphabet: A, B, C, D, E, F. C comes before F. So C is going to be closest to the arm. Okay, so this one's pointing to the inner one. So this is going to be your coarse adjustment knob. Not now, knob. Okay, and this is going to be your fine adjustment knob. Okay, so that's page 1. Okay, now go to page 2. Okay, so now I'm basically gonna help you guys fill this out, and so you could use Google or what I was talking about earlier when I was going through the notes to fill this out, but I'm just gonna type through. I'm gonna type through it with y'all. Okay, so the base. This is basically microscope support. Okay, so then we have the arm is the part connecting the base and the head of the microscope, which would basically be the top, the like eyepiece, the objectual lens. Okay, so part connecting the base and the headpiece. Optical lenses, okay, to magnify the image of an object by using light or electrons. Okay, the stage, oh, where the specimen is placed for viewing. Oh, thanks for hanging in there with me, guys. I know I am kind of all over the place. It's weird. Okay, so fine adjustment knob is going to fine tune focus. Coarse adjustment knob is going to move the objective lens up and down. Okay, light source, illuminator within the base, diaphragm. So I also want you to write right here that this is also called the iris. Okay, it's just another word for it. It controls the amount of light that reaches the specimen. Okay, condenser is the lens that collects, focus the light. Okay, and then the nose piece holds objective lens. Okay, so that is page 1 and 2 of the interactive notebook. So that's all you had to do today. All right, so I'm going to go back to Schoology and make sure. So you should have watched the Amoeba Sister. That is the microscope notes that I went through with y'all. So now if I go to assignments for the day, yep, we did page 1 and 2. So that is it. So that's all you had to do today. Thanks for bearing with me. I'm going to try to post a video every day of like me going through the slides with y'all. So maybe that helps and so that we don't get behind, because biology is state tested and I just want you guys to kill it on a state test. So I'm so proud of y'all. Keep up the good work. I miss you so much and I will be back February 1st. Bye guys.